a tremendously exciting thing for an R end. And I think this idea of sharing data and seeing how people can use different approaches to get at solutions is really an important one for the field, and we're 100% behind it. So I was asked to speak a little bit about our efforts to try to understand cancer genomes using functional genomics. And I think the place to start is to understand, as you all recognize, that the field of cancer research has really been in the throes of a revolution. Some people have described that this is not the end of cancer research or discovery, but it's sort of the end of the beginning. And we all know that sequencing cancer genomes has taught us a lot. We've confirmed that many of the things that we thought were important in cancer do occur and occur in cancers beyond where they were discovered. But we've learned some new mechanisms behind cancer, including epigenetics and metabolism. At the same time, I think we've also realized that the problem is a lot more complex than we ever hoped that it would be. And that is that although there are some genes that are mutated recurrently at high frequency, the majority of cancers, and this is the non-small cell lung cancer genome published by the TCGA, have what is sometimes referred to as this long tail of mutations that occur at 2 to 3, 4%. And the statisticians many years ago would have said, well, we probably don't have to worry about those because they're not very common. But what we've learned is some of these are actually quite important for the cancers in which they occur, and that, in fact, if you develop therapeutics, say for the ALK oncogene, you can induce clinical responses in patients. And so we can't simply ignore things because they're at low frequency, but we have to come up with ways to understand how to make sense of this. What's not presented here is that there's an even bigger problem, and that we find somatic alterations or copy number alterations in a large number of genes of which we don't know their significance. And so one of the problems that we have is we have an increasingly complete parts list, but no instructions about how to put it together or what's important. And that's really the space that my lab has been trying to work in for a number of years. And we've taken a number of different approaches that overlap, but the idea here is that we wanted to have, assuming that we would have this parts list, ways that we could systematically interrogate the function of genes so that we could find those genes that were really important for cancer phenotypes. And as I'll tell you, this phenomena in genetics that's called codependencies or synthetic lethals. Now, to do this, we've taken an old approach, and that is you either take genes or alleles that are implicated in cancer, put them in cell or animal models, and see if you get transformed phenotypes, or do the opposite experiment where you turn genes off and see what happens. And really, our innovation here is not that these concepts are old ones, is how do you do this at high throughput in mammalian systems? And so one thing that we learned very early on is that there are tools that allow you to do this, but what would be really powerful is to do this at scale. And so a few years ago, we initiated this project called Project Achilles, in which we were going to take several hundred cancer cell lines that have been characterized by my colleague Levi Garraway in the Cell Line Encyclopedia for mutations, copy number alterations, gene expression, what have you, and then do loss-of-function genetic screens to look for essential genes, with the idea that this would allow us to have a data set that would allow us not only to understand what genes are essential, but the context in which that essentiality occurs. So after a lot of effort, we've screened a very large number of cell lines. We will probably have probably 550 cell lines by the end of this year. This sort of reflects a little bit two things. One is cell lines that you can get, and second, cell lines, the cancers that affect humans. Now, this is one of the few audiences where I don't have to justify the scale of this effort. Usually I speak at cancer biology or molecular biology conferences, and everyone tells me, well, why don't you just do a few, like 3, 10? And then you'll be done, because why would you need to do hundreds? And so I'm really happy to be here, because you're not going to ask me that question. But I do think it's important to understand some of the reasons why. And I think part of the reason here is, here's an experiment where we took, after screening a lot of cell lines, shRNAs in which we know the answer. The gene is essential when there's a mutation in either BRAF or KRAS. And what you can see is that if you screen 1 or 2 cell lines, you get the answer right 60% of the time. And if you screen 8 or 9 cell lines, you get the answer right 99% of the time. Now, that could be because there are technical issues, and I'm going to tell you that there are not that many technical issues. But really the issue here is that cancer biologists like me are simple-minded. We think that cancer cells are like yeast, and there's a mutation in one yeast strain, and there's not one in the other, and we can screen one versus the other, and everything else is the same, and that's how genetic screens in model organisms have been done, and they've been very powerful. Cancer cells are not that way. They have hundreds or thousands of mutations, some of which are shared with other cells that have the mutation you care about. And when you screen 1 or 2 cell lines, you just conveniently forget about those other mutations. And you really need lots and lots of them to get there. And so what was nice about this is that we had done a back-of-the-envelope calculation before starting Project Achilles and came out with a number that 8 to 10 cell lines should probably give us the power to distinguish these sets. So in this case, it was very reassuring to us that our theoretical calculations were actually backed up by the experiment. Now, the other thing you can do with large amounts of data is you can use the data to tell you about the underlying biology and even how good your reagents are. And so Aviad Cherniak, who's here at the conference, developed a methodology in which you could look at the behavior of all the shRNAs in the entire collection screened over many, many cell lines and use that information to understand which shRNAs were likely to be on target because they would move in the same direction in the same cell lines, whereas the ones that were doing other things would go in other directions. And this algorithm, Ataris, has been quite useful in that regard. Now, RNAi is a very powerful technique, but it's not a perfect technique. And it's important to understand why that's the case. So here's what we're trying to do. We're trying to make this complex that involves AGO2 and dicer to find mRNAs with sequence specificity and degrade those mRNAs. That's the intended idea of RNAi. The problem is every RNAi reagent, whether it be a short hairpin RNA or an RNAi reagent, has a seed sequence, a shorter sequence, which can also enter in this small RNA pathway as a microRNA mimic. And so it can induce microRNA effects. And there's nothing that one can do about that. Everyone in this field has spent years trying to peer at sequences and say, can we just engineer these out of these reagents? And the answer is you can't, because it is part of the sequence that you're using to make your RNAi. And so what Aviad has and his team has done is actually to embrace this on and off target effect and say, well, these are all sequence-based effects, so we can prospectively assign all the reagents in the collection to either be gene-specific or seed-specific and run analyses to take into account both of those phenomena. And what this new algorithm that that was initially called Ataris 2 is now called Demeter for the goddess of seeds. We're hoping that she doesn't give us very many, but whatever the case. You should be able to assign for every shRNA in the collection screened in this way a particular value for both the gene effects and the seed effects. So we've now looked at a number of these, and here's an example of a gene BCL2L1. There are 5 shRNAs we can do. We can see how well they knock down the gene. And what you can see here is that the gene-specific effect are the green ones. These guys knock down the gene pretty well. The ones that don't knock down the gene don't have very much gene-specific effect. But you can see that there's variable amounts of seed effects in these. And with this information, you can be much more confident that the phenotypes that you're looking at with the shRNAs are due to gene-specific effects rather than seed-specific effects. Now, if you're interested in seed space, you could, in fact, do the experiment the other way and look at all the microRNA effects, and there are people who are probably interested in that as well. But I think this is a really powerful way of using the data to tell us about how good the reagents are and allow us to then understand how the data set puts together. So how do we use this? Well, one idea is that you can take all the genes that are amplified in a particular tumor, find cell lines that have those amplifications, look at the Achilles data and say, well, which genes in those amplifications are essential for transforming phenotypes? And when we've done similar experiments like that in ovarian cancer, Tony Chung in my lab found that ID4 is a gene that's amplified in 22% of high-grade serous ovarian cancers, is essential in those cells that have the amplification. And when you overexpress it, transforms those cells. And so it has all the characteristics of what we would call an oncogene. And I would say that this allowed us to get to this answer in a much faster way that would have probably taken the field years to do. Certainly someone can do now in a matter of weeks and months. Now, the other thing that one can do is we've been doing this experiment where you say, well, we're looking for something where there's a mutation or an amplification in a gene, and we're looking for a dependency. And that's how we found ID4 and other oncogenes. But the way this turns out is that that's usually one of the top genes that scores, but there's many other genes that seem to be essential in the context of having one alteration. And this has been a concept that Steve Friend and others popularized many, many years ago, that you could use the genetic concept of synthetic lethality in this context to find codependencies, which are important to think about. And so we've been trying to use this data set to move down this path, and David Barbie, using a very early set of the part of this data set, looked for cells that had mutant KRAS versus wild-type KRAS, and through a number of primary, secondary, and tertiary screens, came out with this molecule, the serine threonine kinase, TBK1, which showed this characteristic that whenever the cells depended on KRAS, it depended on TBK1. And mostly those were cells that had mutant KRAS, but occasionally there were cells that had mutant KRAS that didn't care about KRAS, and they didn't care about TBK1. And then there were cells that depended on KRAS even if they had wild-type KRAS. And so this is exactly the prediction for genetics, that these are genetic relationships and a codependency. Now, we've gone on, and he's gone on to show that the mechanism of how this works is that TBK1, which is normally operating in immune cells to respond to viruses and other challenges, turns out to be downstream of a RAS effector pathway run by RALB, and that RALB directly activates TBK1. TBK1 can activate a number of cytokine signaling pathways, and it turns out that these cytokines feed back on the tumor cells through JAK-STAT signaling and promote survival of these cancer cells. And that what happens when you shut this down is that KRAS-driven cancers don't survive because they don't have this survival signal. And that what's very nice about this is that there'd been a lot of literature suggesting that CCL5 and IL-6 and IL-8 were important in RAS-driven cancers, and most of us said, wow, those are really cool experiments. We have no idea what they mean, and so we just ignored them. And the answer is that, in fact, this is a signaling network that probably affects both the tumor and the microenvironment around the tumor, something that's become appreciated as very important in cancers. Now, there's a small molecule inhibitor that happens to have the characteristic that it inhibits both TBK1 and JAK kinases, and that shows very good efficacy in animal models, and clinical trials testing this concept started about a month ago. So hopefully in the next 6 months to a year, we'll have a sense of whether or not this strategy works. So more generally, though, we're interested in the question of how can you push this? And so you could certainly look for mutations, and we're doing a number of those kinds of experiments. But one other question is, you can divide the data set any way you wanted, based on lineage, based on phenotype, based on the activity of a pathway, and you should be able to find similar kinds of codependencies. And so Steffi Rosenblum in my lab did this experiment by measuring beta-catenin activity, a pathway important in colon cancers and other cancers, and looked in about 100 of the Achilles cell lines and asked for the question, well, what genes are important in cell lines that have activation in this pathway? Now, we found 177 genes that were important. Beta-catenin was the #1 gene, so he was very happy about that. And so what he did was he had this data, he looked at the 177 genes and realized that 6 of them were all related to this molecule called YAP1, yes-associated protein 1, which we knew was a transcriptional regulator that plays a role in many signaling pathways, including the hippo signaling pathway. And what Cepheid ultimately showed was because these guys in the literature were all known to be interacting with each other, that YAP forms an alternative complex with beta-catenin that drives a different gene expression program that's probably important for colon cancer progression. And so this was a very nice example of how you can find a codependency which allows you to map out a part of a pathway that wasn't known before. But I will say that part of this is that he was able to do this because we knew that these genes were related to each other, and I'm going to get back to that in a minute. So if you were to just take a 30,000-foot view of what have we learned so far for an Achilles, and we're still, I think, very much in the discovery phase, is that you can categorize what kinds of dependencies we're finding. There's the oncogene addiction paradigm that Andrea talked about, and that's one. Actually, it's a very strong one, but it's a minor component of what we find. There are things that I'm stealing Andrea's term, a master regulator. We find those, and there are transcription factors that run programs that are very important. And I actually think that that's much like leukemias have mutations in transcription factors and things that drive proliferation. We're going to find the same is true of epithelial cancers. And I've just told you a little bit about synthetic lethals. And so I think it's very useful that we think about these because they represent different ways of understanding the biology of cancer, but also ways of thinking about it therapeutically. Now, I introduced this concept that there's off-target activity of RNAi, and that part of the way you can get past this is to make lots and lots of reagents and screen lots and lots of cell lines, and certainly we're doing that and making the data available. But probably many of you have heard that there's a new technology in town, and that is something called Cas9 CRISPR technology. This is a discovery that bacteria make a molecule Cas9, which allows them to degrade invading viral DNA. And it turns out you can express this protein in mammalian cells, feed it small RNAs, and it will go and make small deletions in genes. And so this is very exciting because it allows one to make true deletions of genes. If you go to any molecular biology conference, everybody's gaga about this, and this is the only thing anybody wants to talk about. Now, we started to play with these reagents, and we made some reagents against our favorite genes. And what we found is, much as what has been reported anecdotally, is that these seem to be much more specific, and they're very, very effective in making deletions of genes. And so it does seem to be quite an interesting question. Now again, molecular biologists we're the kings of anecdotes, and what we really wanted to do was to say, well, can we measure what happens at large scale and really understand how these things work and where they're useful? And so on the way to that path, we did this experiment where we made 2,000 guide RNAs targeting about 200 genes, and we picked 177 genes that came out of our Wnt screen and introduced them into a cell that expresses Cas9, in a pooled format, let the experiment go for a month, and then quantified the expression of the guides in the cells. Now, this is what the kind of data you get, and it's a busy slide, but I'm going to make a couple points. One is the blue dots here are all the controls. And what's remarkable for anyone who's looked at RNAi data compared to CRISPR data is none of those blue dots actually go below 0, meaning that they actually don't have any effect. And so this is really clear that CRISPRs are much more specific in terms of looking at the negative controls. What you can also see is that in all the situations here where we know that the gene should be dependent in particular cell lines, these are 11 cell lines across left to right, we can find that the guides induce, you can see the dependency very clearly. And so if we look at the whole 177 genes that came out of our RNAi screen, what you can see is that about half of them are recapitulated using CRISPR technology. So what that means is that many of the things that you find by RNA are real, and that is also what people have found in the field, but many of the things you can't find if you use CRISPR technology. Now, there are a lot of reasons for that. Some of that it's off-target effects of RNAi, but some of it is due to the fact that CRISPRs are making knockouts. And when you make knockouts, essential genes cannot be studied because essentially they just disappear. And so there's more work to be done to understand that. So emboldened by this, we've decided that the next phase of Project Achilles should be let's screen several hundred cell lines that we've screened by RNAi with a genome scale CRISPR library, because then we'll have the data to really understand how these technologies are complementary and how they are different. And so, whoa. Okay. PowerPoint doesn't like that. Okay. Evidently, PowerPoint doesn't like CRISPRs. Okay, so we're now taking a very large genome scale guide RNA library. It's a second generation Gecko library that was originally developed by Feng Zhang, and screening, our plan is to screen a couple hundred cell lines over the next year. All of them will be screened by RNAi, and that will really allow us to have data that will allow us to compare the two. Now, what I can show you is that we have, as of last week, data from the first dozen cell lines. And here what you can see is these are replicates, and you can see that the replicates are very, very tight. We've done this in very high dimensionality to figure out exactly how we should screen the rest of them. And so this is much, much better than we see in RNAi for replicates. And here you can see the negative controls and the experimentals in two different replicates, and you can see that the correlation coefficient is actually quite tight. And one of the things that you can see is if you take the genes that were essential in these cell lines and do gene set enrichment analysis, what you find is many of the pathways that involve biological processes that you would say are cell essential. And so it does allow us to find things that are cell essential and suggesting that there are real knockouts. So I'm really excited about where this is going. I think the screening process is exactly the same methodology that we use for Project Achilles, and so I think the technical hurdles are not going to be insurmountable. But I think this will be a very nice complementary data set that will allow us to get at some of these questions. And the off-target effects of CRISPRs are likely to be less severe, but they're going to be off-target effects, and we just need to learn and understand what those might be. Now, in the time that remains, I just want to talk a little bit about this other side of the equation, and that is the other way to think about this experiment is instead of doing loss-of-function genetics, why don't you make all the alleles of things that are mutated and ask the question which ones of those are important for cancer phenotypes? And so together with the Orpheum collaboration and Mark Vidal's group, we've made such a collection, and I'm going to just show you an example that was done by Gavin Dunn in which he took the 1,800 genes that are recurrently amplified in high-grade serous ovarian cancers, introduced them into a model where he can, in a pooled format, where we can assess whether these genes induce tumor formation. And I should say here, this was an experiment that was not technically impossible, but monetarily impossible before next generation sequencing. It would have cost $1 million to sequence things using old technology, and it cost about $10,000 in new technology. So, but what you see is that this kind of experiment leads to this result, and that there's a jackpot phenomenon, that the tumor has one allele that just takes over the tumor and forms tumor formation. And in this case, he found that GAP2, a adapter protein in receptor tyrosine kinase signaling, acts as an oncogene. So together with Gadi Getz's group, we said, well, this is great, but don't we have the common problem in all genomic studies that you do some analysis and you have a list of 100 or 200 or 1,000 genes and you'd like to know something about their biology? And so we said, can we think of a platform or approach that would allow any list to be fed into a process, and at the end you'd get a lot of information about all those alleles? And so, for example, the analysis of the 21 cancers that have been sequenced by the TCGA has led to a list of genes that are recurrently mutated at a certain level. And the question is, which ones of those induce tumor formation? Now, we could assign each one of those alleles to a postdoc or grad student around the world and wait 10 years and we'll get the answer. And I think all of us are too impatient to do that. So we've created this thing called the Target Accelerator, and the components of which are this. We'd like to be able to have all the reagents, whether they be mutant ORFs, wild-type ORFs, shRNAs, CRISPR reagents for each of those things. And as I think I've told you, we now have the ability to do that, and we can make about 500 mutant alleles in about a month using this approach. So we have all of those, and then what you'd like to do is be able to take any list, take those reagents, and understand what happens when you perturb the gene by looking at gene expression space, look to see in cancer if that forms tumors in animals. And then since we have ORFs and wild-type and mutant ORFs, we'd like to do a rapid proteomics approach to understand whether protein-protein interactions are altered, and we've done this with Jake Jaffe. And so we're running through these kinds of experiments. We've taken the PanCan list and have already done this experiment. And one of the nice things is we've rediscovered all the known cancer alleles. We found new alleles in genes that are implicated in cancer that are also transforming. A big question that the TCGA has always had to deal with. We have a mutation in RAS at a not 12, 13, or 61. What do we do with that? Well, we know which ones are transforming and which aren't. And with the gene expression and proteomics data, we have a very good sense of what pathway almost all of these guys at least perturb or touch. And I think that if we think about it, that's a great starting point for thinking about which genes to pursue and how to place them in the biology. And so Target Accelerator is in its sort of kicking the tire phase, but I think it's something that I hope will be useful as we think about going forward. And so I just want to finish here and say that I'm really excited about where we're going. One of the challenges that we're going to have is that I'm not a mathematician or a statistician, but I'm convinced that the only way we're going to understand this data is by taking rigorous computational approaches, and that, as I said in the beginning, I think it goes far beyond what one laboratory can do. And it really is these kinds of efforts that people take data and chew it and play with it and try crazy outlandish things that's going to allow us to make progress in deciphering the best ways of understanding what is understandably complex and incomplete data. But I wanted to finish by thanking the people who've done the work. I've mentioned people in my lab who've pushed projects across the goal line, but I also want to say that all of this is operationalized by the structures that we have at the Broad that allow us to do high throughput biology with teams of scientists. And I want to point out that Aviad Cherniak, Sarah Howell, and Barbara Weir have been really driving all of our informatic efforts around Achilles, and they've been also helping to coordinate the DREAM Challenge activities from the Broad side. And I also wanted to point out that Yashashi Shrestha and Jesse Bohm have really built the Target Accelerator, and everything I presented is really their work. And with that, I'd be happy to take any questions before we move on. Thank you very much.
I'm very curious about this convergence of these mutations. So basically, you're now working at the level of genes. If you start thinking back at sort of regulatory mutations that could also affect the expression levels of these genes or the regulation of these genes or the relative balance of, gas pedals and brake pedals and so on, that heterogeneity becomes, extremely large. And what I'm curious about is, at the level of an individual patient, we're often thinking about this series of driver events as opposed to sort of combinations of events. And at the level of genes, we're again thinking about this one driver mutation, versus the synthetic interactions that you showed. So my question is basically, how do we extrapolate from what you showed at the gene level to sort of the network level and the regulation level and sort of go beyond protein coding alterations? So as you're doing your screens, you're probably coming through things that are just not explained by protein coding alterations. So could you comment a little bit about that?
Yeah, sure. So I think we're going to hear a lot about this, and I think you're touching on something that I think the DREAM Challenge teams figured out. And we talked about it a little bit last night, and you'll see the data, but most of the teams actually ended up using gene expression as the most important feature that allowed them to predict dependency. And I think that there are a lot of reasons why that could be the case. There's some technical reasons, but I think it's in part because gene expression is ultimately the summation of all the things that affect what happens to that gene, whether it be mutation, amplification, epigenetic alteration. And so I think that that's telling us something there. The second thing I think I'd say is that we don't know yet what the landscape of regulatory mutations are. And my guess is, we've had a couple hints from, TERT promoter mutations, that's just the tip of the iceberg. And that, one way of thinking about it, and I talked with Miles Brown about this a few weeks ago, and Miles, who's never unsure about anything, said, well, any gene that's overexpressed in cancer probably has a promoter mutation. And it's not a crazy idea, but we just haven't looked because it hasn't been feasible. So I'm 100% with you. Now, the more important question is, how do you extrapolate? And I think the answer is you can't extrapolate from the data we have now. And we have to start, now that we've been able to do these experiments, start thinking about combination experiments. And that's really where I've been trying to head for a number of years. And we're now, I think, poised to do that with the combination of CRISPRs and RNAis and ORFs to do those combination experiments. We started with drugs as a way of doing it because it's an easy way to start thinking about it. But that's really, I think, the future. And that's, but I don't think we can extrapolate very well because we just don't have enough data.
Thank you.
First, thank you for giving the phenomenal talk. I'm just curious to know, is there any time series data in this project that can actually measure the footprint of the evolution of those cells?
Yeah, so the answer is in doing this, figuring out the conditions in which to do the screen, we actually do, we spend a lot of money and we do 10 or 15 replicates and we do many time points and then we pick one because it's way too expensive for us to do that for hundreds of cell lines. But there's data there and there's really interesting changes that occur if you look early on and later on. I will refer you, there's some work from Toronto, Ben Neal's group. It actually looked a little bit with RNAi early versus late, and they found differences there, and so that's one way to look at it. But we haven't really carefully studied evolution of these cell lines, in part because, it's a time and money equation, and you make decisions early on in these big projects. But it's not because it's not an important question.
Okay, so don't leave yet.
Nope.
We, on behalf of the ISCB, we want to give you this certificate for keynote. Yeah, it says for exceptional service to ISCB. And Andrea, we forgot to give it to you yesterday, so. Thank you, Bill. That's excellent. Belated certificate for Dr. Kalipan. Exceptional service.